Historically, we have looked at cancer as diseases of organs, of the lung or the brain or of other organs. But cancer is not really a disease of organs. A cancer is a genetic disease. And I think what we're trying to do at UNC is to, for the first time, really look at the disease itself, the disease of DNA, and take those broken pieces of a patient's tumor, the altered DNA, and design treatments that really target the problem, which is that this is a genetic disease. We're really focused on transition as much as possible to personalized medicine. The goal is to move away from treating a disease called lung cancer to in a direction of identifying what types of cancers our patients have and directing therapy specifically at types of lung cancer. We're doing this in basic science, both through grant-funded research and through our involvement in the large cancer profiling projects such as the Cancer Genome Atlas. The Cancer Genome Atlas will be doing squamous cell lung cancer profiling starting this year. We'll be looking at thousands of patients with squamous cell lung cancer and trying to catalog all the important genomic alterations in squamous cell lung cancer. So the idea is that we take those alterations that we find from profiling tumors, and this is done in collaboration with many other institutions around the country, and then take those basic science findings and then bring them into our own labs and start looking for markers that correlate with clinical response, such as specific therapies. We're also very interested in taking those basic science findings and turning them into lung cancer diagnostics. So the Cancer Center has developed an institution called the RAM Lab at UNC, which is the Rapid Adoption Molecular Analysis Lab, where we can design an assay that is appropriate for clinical specimens, sort of translate those research findings into the setting where we can offer these tests in clinical trials first, and then to our patients in as quick a fashion as possible. That has a technology component as well, so we're looking to sort of newer technologies which may be more robust than what we've had in the past. And some of this work involves having a better understanding of what the standards have been in the past. We had, I think, a really successful project over the last year. They got a lot of national press this summer and has already been incorporated into lung cancer curricula, looking at the reliability of standard diagnostics in lung cancers. What our study did was look at 100 lung cancers and have them reviewed by 24 pathologists, and ask these pathologists to categorize these tumors as much as they possibly could by looking at them under the microscope. And we found that for about half of them we got a lot of agreement in what they thought about the appearance of the tumor and what that meant in terms of the diagnosis. But for the second half of the tumors, we found increasing amounts of disagreement between pathologists using standard techniques as to exactly what category of lung cancer this was. And what this disagreement shows is that we really, not only, and we know this, but I think we need the proof to take us to the next level, that standard techniques are not going to be sufficient going into the future, or at least they're unlikely to completely describe the diversity of these tumors. And that really reinforces our need for something like the RAM Lab and these new biomarkers that we're working on. And it helps us to benchmark how we've done in the past so that we know as we go forward, where have we made improvements and how much improvements have we made with these new diagnostics that we're working on in terms of things that have been suggested by our basic science and from our clinical and translational research. So the idea with these diagnostics and this understanding of basic science is then to take that forward into our therapeutics. We have trials in the first line. We have trials for patients whose tumors have come back. So we've certainly got a wonderful collection of trials for those patients. But then we also have trials that are really somewhat revolutionary in terms of targeting these new and developing therapies, and one that we're particularly excited about. It takes a target that's only been known for a limited amount of time in lung cancer, a translocation or an alteration of a gene that's present in normal cells to make a tumor-producing gene. Well, this gene is known to be present in a small number of lung cancers. There was great evidence from our large research consortium presented this summer that there's a therapy just specifically for that that altered gene. And so over a very short period of time, we've taken this novel alteration found in lung cancer. We now have a biomarker for it in terms of a test to test for that alteration, and a therapeutic. And we're going to be opening this trial, in fact a couple of trials that target this gene over the next year. And so that's a really exciting pace of development. Now this gene is only altered in a small fraction of lung cancers, but those patients are really approaching a truly personalized therapy for patients who have that tumor alteration. So I think that's what's going on nationally, but it's, I think, very well reflected in what we're doing here at UNC.